So the difference between a gene and an allele is that a gene is really the thing that makes the protein. It's the instruction that makes a protein. However, an allele is like an alternative gene, and so you have typically on your chromosomes, you may have on two different chromatids here. You have your gene that's basically specified in both of these at the same level at both of these on these chromosomes at the same side of each of the chromatids here. But the allele is where one copy of that gene is slightly different than the other. And so you have this difference here. So I'll just read this. A distinct sequence, a gene is a distinct sequence of nucleotides forming a piece of a chromosome. A gene is a sequence of nucleotides in DNA or RNA. So it's not just DNA, but it's also RNA. But it does something. It creates some kind of protein which has some kind of function. Now, whether or not that function is known, that's entirely something else. But it does create something. It has a product. It's a coding thing. It creates something. It's a piece of computer code, if you're thinking in terms of computer code. But it does something. But the allele are, for instance, two competing functions in that computer code. So it's like, and they both do the same thing, and which one you choose is entirely random. And so, for instance, the allele is an alternative form of a gene, you may have a function here in a piece of code, and then you have an identical function later on, and you never really know which of those functions in the code is actually chosen, is actually used. And so your allele is an alternative form of that gene. Excuse me. So here we have the genes versus allele. So what I was talking about is here we have a chromosome, and the bands, the red bands and the yellow bands, that represents a gene. And so when you are reading, or when your body is reading, or your cells are reading your chromosomes to get the DNA out and to make proteins and all this, you have BB, which is the gene that it encounters. Now, typically, your gene is going to be BB. In this case, and the first one is BB. That's your gene. Or if it encounters big B, little b, and I'll talk about what these mean. But the B's here are just names that we use for the alleles of the genes. And so here we have BB, big B, big B. That is the gene. And big B, those are two identical copies of the gene. Big B and big B are both what they call dominant genes. They're both alleles, but they're both copies of the same gene. Now, going to we'll talk about these terms in a second, homozygous and heterozygous, but going to the right-hand side where you have big B, little b, this is a kind of a descriptive way of saying that I have the gene, big B, little b, but I have two copies of that gene. I have the copy big B, and I have a copy little b. And so which copy is being used depends on the genetics. But typically what we would say is that the capital B is going to be what they call the dominant copy of the gene. That's the copy that gets read, or if you're creating that protein, then you go the route that says, give me the protein from the big B copy, not the little b, but the big B. And so big B, or B, or big B, like we have on the left-hand side, or big B, little b, those are what we call the dominant, or the big B is the dominant allele. The little b is called the recessive. So that brings me to the terms homozygous. A homozygous is where you have, for instance, on the chromosomes, you either have the big B, or you have little b, little b. In other words, there is no variation. It's the same copy of that gene on both sides of the chromosome. Heterozygous is where you have a dominant copy of that gene on one side, and the other side you have a recessive copy. Now, when I say recessive, that means that it's the copy that usually isn't expressed. It's not typically expressed. It's usually the dominant gene that's expressed. Now, the reason why you have to worry about the dominant and recessive is because you tend to have genes that work out that way, where a dominant gene is the one that gets expressed, and the recessive one is the one that kind of is dormant, and then that's the one that gets passed on. But if you remember that, for instance, your genes and genes are the things that give you proteins, the alleles are the differences in those genes. Now here we have chromosome 1 and chromosome 2, and you can see that allele 1 is the big A, and allele 2 is the little a. And you can see on the bottom line here, I don't know if you can see where I'm pointing here, but hopefully you can't. Well, I guess you really can't. maybe here. So these down here, those are the chromosomes here, which are the inheritance. You know, are inherited from the chromosomes allele 1 with allele 1 and allele 2. So you can see that there are 4 possible types of chromosomes that you can get. In other words, this is where the probabilities come from that we began to talk about last class, where, for instance, you may have 4 children, and one of those children will have, or is likely to have, not necessarily, but it's expected to have big A, and then you have 2 children out of those 4 who are expected, when I say expected, I mean statistically expected to have big A, little a, and another child is expected to have little a, big A, and that makes half of the 4, so that's 2 of the children have those 2. And then you have one of the 4 children is going to be little a, and so these big A's and little a's are inherited directly from the parents, and so that is really the essence of what we're talking about today, and that is that you have these genes that are inherited. Now, this is actually what they call Mendelian genetics. And when I first saw Mendelian genetics, I was flabbergasted. I thought this was actually one of the coolest things in biology, and that is that there was a priest who lived some time ago, and he was working with one of his experiments he worked with were these plants called the Pisum sativum plants, which are basically pea plants. And he noticed that some of the plants, he also worked with flowers, but he noticed that these pea plants, though, he noticed that some of the plants had round, full peas, and other plants had kind of shrunken, kind of dry-looking peas as products. This he recognized was some kind of a trait. It's just like a genetic trait. And so he thought that maybe there's something going on. And so he began doing some research, and he began to interbreed the plants that had the round, full, or as he called them, the big S variety of peas, with the plants that only produced these shrunken, dried-up looking peas, which he called little s. Now, he noticed that when he began to interbreed these plants together, he noticed that the plants fell into 4, basically, or actually not 4 different categories, but he noticed that a fourth of the plants, statistically, and he did this many, many times, but a fourth of those plants tended to have the shrunken peas as the fruit from the plants, just a fourth of them. And so then he began to realize that perhaps there was this, maybe there was a trait here. There was some kind of a trait that was being inherited, and you could kind of predict the statistics of this trait based upon this what's called the Punnett Square. And so with more experimentation, he came up with the whole notion that each gene has a backup copy. And I mean, this is his model here, his working model. And so he said that each plant has either big S, or little s, and that makes it full round or shrunken. And when you interbreed these things, then you get some traits that could happen. Now that's not very telling, but this is maybe a better example of what's going on. Is that when you use a Punnett square in this case here, you could use peas or you could use flowers. And in this case here, on the left-hand side, I'm looking at beans. And you find that there's a big Y and little y on the, going on the vertical line, and horizontally along the top you have little y. And this is really how the Punnett square works to kind of cross things over. And so what I'm doing here is I'm talking about when you have a generation that comes out of 2 parents, then the generation that comes out has a genetic makeup that looks like the makeup in the Punnett square. So I can't point at anything here, which is unfortunate, but I'm looking at the left-hand side Punnett square in the upper left corner, and you see a big yellow Y and a little green Y. Well, that's because you get from the Y on I just call the Y-axis here, but on the Y-axis you get that yellow Y, and on the X-axis you get the little y, and so that statistically, the offspring would be a fourth of the offspring would get that particular makeup of genetics. So that's like a statistical expectation. On the other hand, because one of the parents here is little y, then on the right-hand side, on the upper side of this Punnett square, statistically another fourth of the generation would come out of would be made up with big Y, little y. And so really in your whole population of things here, you'd expect to find half of your population with a big Y, little y makeup. Similarly, when you look at the little y on the x-axis here, and you go down to the very, very bottom of the Punnett square, you'll find that the only thing that offspring can get genetically is little y. And the same thing if you go to the right-hand side, you'll find that offspring is also going to get little y. And so when you start doing crossover experiments, like generation after generation, and you start looking at the numbers of populations and you kind of figure out what kind of visual trait they all have, you'll notice that you have half of the group is going to have the trait that comes from the yellow Y, and the other half of the trait, or the other half of the group, is going to have traits that come out of little y. And in this case, little y is going to be the recessive, and the big yellow Y is going to be the one is the gene that actually expresses itself. So that's if you're working with peas, but you can also work with a number of other things like flowers. You can also cross-pollinate flowers, and you'll see this isn't the best example here, but you can see that you can mix a pink flower with another kind of pink flower, and you'll find that after a while, after some generations, you'll find that a fourth of those flowers would come up with you'd have a white flower, and that's if, for instance, the parenting or the parenting flowers have a recessive allele in them for white. In this case, white, little b is a white flower, and big B makes a pink flower. And so again, you're able to kind of look at the numbers of flowers that come out of this and say, of all the population, and the more flowers you have to work with, the better, but of all the flowers that I have, I'm finding that a fourth of those flowers are white. And so that suggests to me that there must be some kind of statistical reason for why those flowers are becoming white. And this statistics here helps to explain that, where you have one parent is big B, little b, where big B is the pink color flower and little b is the white, and the other parent is the same makeup. And when you have this Punnett square that looks like this, you can see that the flowers are becoming, you can see that you get these white flowers, which is unusual though, because you don't see white in the parents. And so that suggests that the white gene was always there, but it just wasn't being expressed. So that's quite exciting. Okay, so now where are we going with this? We're actually talking about the cystic fibrosis gene, which has a very similar kind of makeup. This is actually a much worse. This is a much more urgent type of research, I would claim, in bioinformatics, because it actually affects health. But you'll see that the makeup of the cystic fibrosis gene transfer, or the inheritance, is very similar to working with Punnett squares. So the cystic fibrosis gene is really something that's responsible for making a protein which is what's called a bad regulator. That basically means that it's like a piece of the cell that's not doing what it's supposed to do. Imagine, for instance, if you're driving your car and the pump that's supposed to move the fuel from the fuel tank into the engine wasn't working, then you'll find that that pump isn't working, then the whole car is rendered useless. It's one small piece which actually has a crippling effect on the rest of the whole. And so this is exactly what we're talking about here, and that is that there's a protein product which is at the cell wall, and that protein is supposed to move these chloride ions by osmosis into and out of, and around in the cell here. But the thing is, though, that as it's moving these chloride ions around, water is able to be displaced. That's how it functions. It has to move these ions, and then the water is able to move at the same time. But water flows or follows these chloride ions by osmosis. Now, imagine, for instance, that you're unable now to remove these chloride ions. That means that the water isn't going anywhere, which means that things could be bad. So let me just ask you something here. Imagine that you're unable to regulate the water in your cells. Could that be bad? Is that a terrible thing? If you think about how much of your body is made up of water, I mean, it makes sense that you really do need to start being able to move that water around to different places. I mean, managing water in your body is essential to health, absolutely essential. So how much of the body is made up of water? It's about 90%. It's about 90%, and so it's a lot of water, and so you need to be able to move that around. And so cystic fibrosis is a genetic disorder where, for instance, water is not able to be moved around in areas like the lungs and in other places. And the thing is, though, that when you don't get water moving around, then, for instance, you get the mucus begins to build up inside your cells, which is, or actually around the cells, like in the lining of the lungs. You don't want mucus to build up. Oops, sorry about that. You don't want mucus to build up in your lungs, because if you have mucus building up your lungs, it makes a place where where bacteria can live, but also more importantly, in your lungs you have these little hair-like things that are called cilia, and the cilia kind of move along in this kind of wave-like pattern, and they actually move out all the pieces of dust and debris that you might breathe in, and that's how you clean out your lungs. These little hairs move things through. But if those are stuck in mucus, they're way down. They can't move. They can't. I mean, it's difficult for them to move effectively when they're surrounded by all this mucus stuff, and so they cannot move, and they cannot move things out. They cannot do their job. And so the thing is, though, that now you have this debris and this dust and all that this kind of keeps accumulating, and that's where bacteria can grow. And so you can get very, 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 very sick. So really, cystic fibrosis just makes thick mucus grow inside your lungs, and that prevents other things from doing its job. Thick and sticky. It's like cystic fibrosis causes mucus to become thick and sticky. This is just another picture of this if you're interested in seeing more about what's going on. But some of the symptoms are, for instance, that you have this is a normal airway inside your lungs. On the left-hand side. On the right-hand side, you have dilated airways which begin to get filled up with all this mucus because the cells are just unable to move water out. Everything becomes waterlogged. And you can kind of get that if you imagine you put on a sweater which is like still, straight out of the washer, it's still soaking wet. It's actually very hard to move around because it's so heavy, but also it's filled with water, and so it's just very, very puffy. Now imagine that happens to the lining of your cells, or in the endothelial cells of your core, or in your lungs, or wherever it may be. Imagine those cells are becoming really kind of just waterlogged and soaking wet, unable to move that water out. Then it's like things can't move around it, and things in your whole the airways become kind of more blocked because the cells are just swollen. So I guess the take-home message here, though, is that a buildup of anything that shouldn't be there, a buildup of any kind of toxin, is very, very bad for you. Things can get very, very messy if you don't clean these things out. And so this is just kind of a disgusting picture of this. This is kind of a horrifying picture, I think, but when you have cystic fibrosis, you get what's called clubbed fingers, where the ends of your fingers become actually swollen. And this occurs when your lungs and your heart are basically unable to process oxygen and the blood as they normally would. And so it's a very serious type of disorder here. By the way, if you're interested in learning more about the blocked ion channels, there are some two videos that I found which I think are actually very good. I think the first video here is a very short video. The second video is a bit longer, where it's talking about the actual ailment of cystic fibrosis, whereas the short video here is talking about, I think they both show the proteins here, but this one here is just showing you the protein. It doesn't actually talk about it as an ailment. But really what it is, though, is this is, in this picture here, this is the inside of the cell. You're looking at the ceiling of the cell, and there's this thing here that looks like a piece of gum stuck to the ceiling, but actually that piece of gum is the port, if you will. It's a port in the cell that lets out these ions, and in doing so, it actually moves water around. And the thing is, though, that if you have a mutant cystic fibrosis transfer gene here, then that means that this port is permanently blocked, and so nothing can move through, including the water. And so therefore, you have this. And by the way, these little white or yellow balls that are in this thing here, these little bubbles, those are the ions. But the thing is, though, that if the protein's not working, the ions cannot move through. On the right-hand side, you see a functioning protein where the ions are moving through, and on the left, it's not functioning. Nothing is moving through. This is just another picture of what you can see here. But the interesting thing, though, is that the whole pump works by just moving salt around your body. And the idea is that by moving the ions out, the water will follow. It's like you're creating this thing. But if you in the bottom of this cell picture here, you can see that in this cell, it's this pink thing, which is filled up with the NaCl, or just table salt, that's sodium chloride, I guess. Actually, not. Yeah, that's your salt. But if you're not moving these chloride ions out of your cell, then the water kind of rushes in, and you also cannot wash away the mucus that's like above the cell. It just kind of forms there, but then it dries out and it becomes kind of a sludge. So anyway, why are we talking about this? This because inheritance for cystic fibrosis works very, very similar to the Mendelian Genetics inheritance that we just saw with the peas and with the flowers. And that is that your mother or your father, or the father of the patient, mother or the father of the patient here, if they are cystic fibrosis carriers, then it means that they have one allele. If they're a carrier, that means they're not expressing any cystic fibrosis, but that means that they have one gene which does express cystic fibrosis, and the other one which does not. And so the thing is though, that they're carriers because they're not expressing that. They're not showing that cystic fibrosis disorder, but rather they do have cystic fibrosis in their bodies. And so one allele is what they call the wild type, and the other allele of this gene, of the gene that produces this protein, miscodes something, or there's an error in there somewhere, which causes the cystic fibrosis to happen. Now, that's the mother and the father, so it seems that they are both carriers here, but when you look at their children, they have 4 children here, and statistically, one of the children is likely to receive both copies of the normal proteins, so that they are not a carrier of cystic fibrosis. In other words, they receive from their parents, each of the parents give a copy of the working gene to the child, and so the child does not have cystic fibrosis. That happens. That happens a fourth of the time, in fact. On the other hand, another fourth of the time is where the child receives 2 copies of the broken gene from the parents. And that means the child on the far right here, this child is receiving 2 bad copies from the mother and the father. That means it's receiving, I think this is a recessive gene, an autonormal recessive type of condition. One faulty gene is inherited from both. So that means that the parents are receiving the parents are giving their recessive bad copy to a fourth of their children. Now, this doesn't always happen. I mean, if you have 4 children, it doesn't necessarily mean that one child is this and the next child is the other way. It doesn't mean that there's like some kind of checklist that each child has to check, in terms of their genetics. It doesn't always work that way. But what these statistics are saying is that, in general, if you look at all of the situations where the parents were both carriers of cystic fibrosis, then of all the children that came out of families with that kind of genetics, a fourth of them did not have they did not have both copies of the bad gene. A fourth of them had both copies of the bad gene as inherited, and then half of those children, in other words, 1/4 plus another 1/4 of those children received 1 good gene and 1 bad gene. That means 1 dominant gene and 1 recessive gene. And those children may themselves be carriers like their parents, and sometimes they don't know that they're carriers. One thing that I've noticed, though, is that when you start looking into like some of these journal papers that talk about spotting cystic fibrosis and those things, some of the papers are saying that, or some of the things I've read, though, have been talking about having more kind of genetic tests for parents or for would-be parents, so they can check their genetics and see whether or not they are carriers of things like cystic fibrosis or sickle cell anemia or other types of disorders, so that they can be prepared when their children are born and they may need some special attention. And so I think that with bioinformatics, though, I mean, there are ethical implications all over that, what I just said. Checking parents for genetics, and that's entirely filled with ethical issues that need to be figured out. But the thing is, though, that with bioinformatics tools, we can actually look at the genes. We can actually take a sample, study the area where there'd be a problem, and see whether or not a father or the mother are carriers of cystic fibrosis, and therefore you can decide at that point whether you want to somehow, I guess, have children, or if you want to maybe, who knows. Again, this is rife with ethical concerns. Now, let's talk about the actual area of like where the cystic fibrosis comes from. Cystic fibrosis, actually, I'll just give you a heads up here. It's a mutation of just one one area, this one base is misspelled in your DNA. And so remember when we were talking about translating proteins from RNA, and that is that RNA is read in triplets of 3. Basically, you're taking your RNA code or bases, and you're reading them 3 by 3 by 3 by 3. And each 3 stands for a kind of it's a code word that codes for some amino acid in protein. So in this case, AUG, that's methionine, which usually begins most proteins, although that's not always a hard and fast rule, but it tends to be that way. The next one is ACG, which is threonine. You have all these different amino acids, which are then kind of, they're all built, they're all placed on a chain according to what the RNA says. And so then at the very, very end, you'll see that there's this stop, which is UUA. That's the end of the gene. By the way, when you're looking at some code to see whether this is a gene, typically you'd see whether, do I have an AUG that starts this thing, and do I have an AA or a UAA or a UAG or a UGA at the end? These are, you usually have one of these 3 that's at the end of these. But when your genetic machinery is creating the product of a gene, it would start by creating this one and go all the way to the end until it hits one of these guys, and then it would say, okay, I just found the UGA. I'm going to stop now. My protein is done. But the thing is, though, that as you're coding along the way, if one of the pieces of the DNA in the middle here is somehow corrupt, or it's misspelled, or a bug somehow got in there, or a neutrino from space hit an ancient relative of yours and knocked out that particular base in their DNA during some kind of critical time, and the base was not corrected, then that's a very serious thing, because that DNA mutation, while it didn't kill the host, it became this mutation which crept into that family, and so that becomes this. That's really where some of these mutations have come from. Just space radiation. That's a strange thing to say, but it's true. Now, this is actually quite interesting. So when we're talking about reading frames here, notice, getting back to this, notice that we're reading our bases in 3s. We start someplace, and we first of all hit methionine and threonine and so on and so forth. We start with AUG, and then we move to ACG, and then we have GAU, just as we've kind of like shown here, we're allotting each codon. But that's really all dependent upon where you start reading. So here's an example. So here we have a sentence: Pam can see the man and dog. These are a whole bunch of words that are 3 letters long. And when we read this, we start reading with the P from Pam. Pam can see the man and the dog. That's how our eye clusters these letters together so that we can make sense of the text. But imagine now that, for instance, I remove the P from Pam, and I start clustering my letters in terms of, or in words of 3. You'll see that you'll see that this word right here is basically 2 words, the pieces of 2 words kind of coming together. I'm suddenly getting gibberish now. Just by removing this one P, I'm getting gibberish, and if I know that I'm reading only words that are length 3, here I'm not getting any sense. It's like I just removed one letter, and suddenly my reading is not giving me anything interesting that I can use. Now let's say, okay, fine, okay, I'm going to remove the A from. I've already removed the P, but I'm going to remove the A again, or the A now, and then start making my clusters of words of length 3. And again, I get gibberish. I can't read anything on this. This is nothing. I mean, it's interesting to see what happens to the text, but in terms of actually delivering a message, you get no message from this. And then you get these other ones that are like left over at the end. You can see that these other bases here are kind of bases which are not part of any codon. So that's too bad. And so then now if I remove the P, the A, and the M, then I can see that suddenly I can get my words back. I can suddenly start clustering my words together in lengths of 3, and I can start making some more sense. I mean, of course, I've lost a word. I've lost the noun. Who can actually, I guess it's called the pronoun. But I've lost the subject of the sentence here. Who can see the man and the dog? It just says can see the man and the dog. So I've lost information here, but at least now I can still get some. I get some meaning out of this part of the sentence here. Ideally, I wanted to have this whole sentence here where I know who saw the man and the dog, but now down here, I have an idea that there's a man and a dog involved. But you can see, though, that by changing how you're creating your words, you're actually changing much about the actual sense of the message here. You're changing much about what you're able to extract in terms of information. Now, let's go to DNA for a second here and see the same thing happening there. Now, I have taken my regular, this is my original DNA sequence here, and I've cut everything up into red and blue words. And if I'm reading my first codon, which is CAA, that is a codon. It codes for some amino acid. I'm not exactly sure off the top of my head what it is. But that's my first codon. My second one is here, third, fourth, and so on. Now, if I remove my C and I start reading at this word here, AAT, the AAT becomes my first codon. In reality, this should have been my first codon. I should have began reading at C, but I didn't. C is gone somehow. And so now I'm reading here and clumping my words together in threes, and that's my codon. This codon may not be appropriate for the instruction code that's coming out of this DNA. And you can see, for instance, that while the words, if you put them back together and regroup them, you can see that you can actually make sense, but the codons as they're read are actually not, they're meaningless. Now, if I remove the next one here is CA. If I remove the CA and I treat ATG as my first codon, ATG, you can see that came from here, but that is totally inappropriate because it has nothing to do with the protein. In fact, maybe that's not even the methionine that I need to initiate my protein here. And so what I'm saying is that you have now, just by taking this off or taking off this, you've actually created this mutant protein because everything is completely, everything's out of register. Now, you can do the same thing in terms of, if you look at, for instance, like how your DNA will actually be read, you're seeing that really your reading frame, if you're going the opposite direction, can also become kind of changed. If I'm starting from this side here, AA, and I kind of remove my AA and I start reading with ATA, or I remove the ATA and I'm starting to read over here. What I'm saying is that when you start pulling pieces off the front of your sequence, it really upsets the entire register of how your proteins are being created. And so that is something about how cystic fibrosis becomes. It's like you have these slight mutations, tiny mutations, which create enormous consequence in genetics. Now, one thing I wanted to talk about though is reading frames. If I can just copy this thing here, I found this online tool which I think is actually quite good. If I go here and I paste this, hopefully you can see this. There we go. So what we have here is an online tool that allows you to input DNA, ATG, 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 ACC. And if I hit translate, what it's going to do is it's going to do exactly what our code that we wrote in class does. And by the way, that dogma code, I've put that into today's lesson plan because I've finished the code that we were working on in class. I'm sorry I didn't mention this yesterday. I forgot to do that, or Wednesday I forgot to do that. But the code is complete, and I do invite you all to look at this code and to kind of see how it works. But it does the same thing as this, really, where it doesn't work with frame shifts, but it translates DNA into the proteins. We have the same functionality coming out of BioPython. No need to call up some big protein or some big protein generation website like this. But what I'm saying, though, is that my gene here is ATG, ATG, ATG. Now, this is what they call the frame shift here. And then by removing my first protein, my first base, I get a completely different protein. And then by removing other bases, I get another completely different protein. And so what I'm saying is I have these proteins here, which have nothing to do with the, I guess what I call the original bases that I used to spell out the protein. In other words, MMMT should have been the protein that I would have got, but when you start pulling off bases on the front, then your reading frame becomes different, and then the proteins that you get are in some cases a complete surprise. And so you can spend some time looking at this. In fact, let me just go ahead and I'll just copy this a couple of times here, make it a bit longer and see what we get. The translation, and you can see that my protein should have been this, where I have, my protein should have been this, where I have all these MMTs. But the thing is, though, that maybe this protein here, G's and H's, maybe, for instance, biologically, I can't have a protein for some reason that has, because they respond badly to other proteins in the neighborhood. Those G's and H's are somehow attraction points between this protein and another protein, and the proteins stick together like magnets, and it causes all kinds of biologically dangerous conditions. And so, or maybe, for instance, when the protein is created with a bunch of D's and H's together, then that becomes a dangerous protein to have in the neighborhood. Maybe that upsets other proteins, and then other proteins can't function. And so what I'm saying there is that by pulling off like the bases off the front here, here, and here, like if you're pulling off the first two bases, you change this reading frame, and that could cause chaos for certain types of protein production. This is actually quite interesting. You're talking about, I mean, this is, believe it or not, we've actually discussed most of this stuff in our overview. And I like to think that in our class, the first discussions that we had about bioinformatics are really kind of like the general ideas, and everything that we see after this, after those ideas, seemingly everything, is really just a matter of examples of the stuff that we know to be true. And so, for instance, we're asking ourselves whether a sequence is a carrier. That means can we get cystic fibrosis from a piece of DNA? In other words, is there an allele for cystic fibrosis in that DNA? And so what you would tend to do is you would get a sample of that DNA. This is how you would test it. You get a sample of that DNA, and then you put it into a protein using a software that's very similar to this, very similar to the protein translation software that you created in this class on Monday. And then you have this piece of protein, and then I know that, for instance, where this question mark is, these diamonds, I know that at that point, if I get one kind of protein over another kind of protein, or I get one kind of, if I get one kind of amino acid over another kind of amino acid, then I know that that amino acid is maybe free of cystic fibrosis, or another kind would create cystic fibrosis. And so really what you're doing in science is you're just taking the DNA, you're creating protein, and then you decide whether that protein has the traits of creating cystic fibrosis. And so I know that this is just 3 lines in this slide here, and it seems very straightforward and very simple. I mean, how hard could this be? But actually, it is very difficult to do some of this work. But luckily, though, we have these machines in bioinformatics which do this work for us, and it can be done seemingly in an afternoon. You can find out all this stuff in an afternoon. But back in the 70s and 80s, much of this work had to be done by hand, and maybe it couldn't even be done back then. Maybe it's even in the 90s. But it had to be done by hand, and even then, it wasn't likely to work because, I mean, your hand may have been shaking as you're doing something, and so maybe it destroyed the DNA or broke something. And so it may have taken maybe weeks or months to actually get an answer. And so anyway, that's what you're doing there. You're looking to see what the DNA does when it makes protein. Now, remember this codon table here? This is where these codons are coming from. And so getting back to this over here, if I see, for instance, where that diamond is, if I see that I have a tyrosine, which is a TAT triplet, where I should be having something like a cysteine, which is a TGT, if I see that I'm getting something that I'm not supposed to have there, then that might inform me that perhaps cystic fibrosis is showing up. So here's basically the summary here that we've been looking at. And by the way, I stole this from your textbook, and so I do hope that you're recognizing that this is actually from your textbook. But this is really talking about like how your DNA is being used to create these proteins, which you can then use as analysis. Now, one thing I'd like to talk about here, which is which actually it's hard to actually get this message from the book, but it's here. I want to talk about the non-template strand and the template strand. When you're doing an experiment where when you're doing this experiment here, which side of the DNA do you start with to actually get that gene to determine whether or not the gene is going to make you sick? And so you have to start with what's called the template strand. Now, here's the thing. When you have DNA, you have exactly two different strands of DNA. It's like the ladder, which has the rungs and it has the side pieces. You have two sides on both DNA. And you have to figure out where the gene is that you're actually going after. And then you have to use the opposite strand is, so that when that strand is complemented, in terms of when it's going through its division and everything, when it's complemented, then you can actually grow out the gene that you need to create that protein. So in this case here, what we're looking at is that you'll have the non-template strand and you have the template strand. The template strand has the complements of the gene as it is. Actually, the gene as it is, as we're looking, would be found on the non-template strand. But we don't take that strand because we want the complement of that, because we take that complement off, that becomes the mold, if we will, into which we form another strand, which is the gene itself. And that's how you copy DNA. So I take the template strand, which as DNA does not contain the working copy of the gene as we can use it, but rather it contains the makings of copying that gene. So we take the template strand, and then from DNA, you get I don't like using the word template again, but you get a working copy of genetic code from which you can create the protein. So look at the template strand here, where it begins, it's the 3' to the 5' directionality, where you start out with, on the 3' end, G, C, T, T. Well, when you copy that code, then that 3' end on the copy is the 5' end. 5' end on the RNA is C, G, A, and so on and so forth, 5' to 3'. And that's how the directionality of RNA works as well as DNA, but that's how you read, that's how you're reading that code to create protein. And so you would start by making a protein with this coding sequence going from the 5' to 3' direction. And in this illustration, what they've done is they've started with AUG. AUG, surprise, surprise, AUG is methionine, ATG. You see on the left-hand side of the screen here. Left-hand, fourth square down, or fourth entry down, that's methionine. And methionine, M, is usually how you would begin a protein. And so here we have the beginnings of a protein. AUG, CAC, GCC, UAU, and so on and so forth, going from the 5' to 3' direction. But the whole point of this is that it's important to realize that when you start to actually create your protein, when you're running this experiment here, you're starting with what's called the template strand, which is a faux ami in terms of a definition of a strand here. It's not really, you can't use this strand, you can't just pull this off the shelf here and then attack or then put your protein creation machinery on the sequence and make protein. But rather, you have to first of all go through this process where you're creating RNA, which then converts the template strand to its complementary and reverses its directionality, and then you can create your protein. And so that is something that I find that is very hard to kind of get through this book. I think that the text is there, but if you spend some time actually looking at this and remembering how DNA works, 5' to 3', finding the proper gene that you want is on one of those two things here, but is it the complement that you want, or is it the regular gene as it is? How do you actually get that gene out and into the machinery that is out of how do you get that gene out of the DNA and into RNA and then used to make protein? Well, you have to start with what's called the template strand, which is the complement of the gene. Anyway, if you have any questions, let me know. I don't want to that's actually a big it's a big thing. It's actually a big piece of it's actually a very big piece of biology, and I wanted to make that absolutely clear. Okay, so remember that this is kind of like how you can remember how this all comes together here. If you get a piece of DNA, this is a very basic algorithm that you'll see in types of DNA research. In fact, in many papers of bioinformatics, you'll see these kinds of scenarios where basically it's talking about the flow of information around the algorithm. It's very visual. But basically, you would start with a DNA sequence, and you ask yourself, is this DNA sequence, or my template strand, in in the right kind of directionality. In fact, is this in the 5 to 3 prime? And that's, you're asking, for instance, is this really, or I guess you're asking whether it's an RNA sequence or something, but is this in the 5 prime to 3 prime direction? If it is, then you do nothing and you continue. And then you translate the protein and you translate and study the protein. However, if the protein you get is not in the 5 prime to 3 prime direction, that's not true. It's not the right direction. It's not the right directionality. It might be, let's see, if it's 5 to 3. If it's 5 to 3, it means that you're working with the non-template strand, not the template strand, but the non-template strand. You can't start with that. And so therefore, you go into then you have to change this into the 5 prime to 3 prime. In other words, you reverse the direction of this thing, and then you have to go through the RNA and the RNA transcription and the translation processes to create the protein. One thing that I haven't mentioned here, though, and I'll say just a little bit about right now, is how do I know where this gene is? How do I know whether the gene is found on what's called the template strand or the non-template strand? Remember, the template strand and the non-template strand are really named in reference to the gene. And so I have to, first of all, find the gene, and I have to, first of all, know that the gene has a beginning and end. And so I have to know something like maybe the gene would begin with the methionine. I have to know that maybe it ends with the tyrosine. I'd have to know something about this gene. And so that's actually wet lab work you'd have to do in some kind of biological setting. You'd have to know that the gene can be grown from this code. And then once you know that, then you have to figure out which is the left-handed and which is the right-hand sequence of the DNA. So once you know what the gene is, then you can determine what the template is and what the non-template strands must be in your DNA to actually create that protein. So that does take some time, and you do have to do some work here. These are just some backup slides that kind of talk about the how you can other ways of reading this same algorithm down here, but template or non-template here. First of all, you have to find out whether the code that you have is the right directionality for the gene. In other words, is it 3 prime to 5 prime, or is it 5 prime to 3 prime? And you have to figure out where it is. Now, when you were looking on Wednesday, when you were looking at all these different tools, and even when we went to Uniprot and looked at the tools online, we'll spend some more time going through this in the next classes to come. But you'll see that your DNA has been annotated. And any pictures of DNA that they have on these websites, the DNA tends to be annotated. And by that, what I'm saying is that you have these kind of blocks on the DNA. You have the 5 prime to 3 prime DNA. These are the two lines that you see right here. I don't know if you can see me here. But those are the two lines you can see. And under those, on those lines, we have these blocks. I'm looking at, for instance, the upper line of this DNA on the 5 prime to 3 prime direction, where I have this large, heavy, dark or black line, which is AG or ATG, all the way through TAA. And those is significant. That's significant because that's a gene or it's some kind of important landmark. But in this case, it's a gene because you're talking about. I mean, it's centered and it's what this. It's a gene here because it's dark. The other ones that are the boxes that are not dark tend to be types of landmarks. Maybe they're smaller genes, but maybe they're something you'd use to find bigger genes. I mean, if you think about it, this is all A's, T's, G's, and C's. And so in order to find a gene, you have to use other pieces of information, and they call those genetic landmarks, where you can find, if you find those, then you know that the gene is coming up soon. It's like it's a primer in some ways to say that the gene is coming. So that would tell you, if you can find those landmarks, you can find that the gene is coming up, and that means then you can see, well, then I have to analyze and see whether this is the right directionality. If it is, maybe I'm working with the non-template strand. If it's this direction here, 3 prime to 5 prime, then I'll call that the template strand for that section of DNA. So in other words, I'm grabbing the gene that's on this side of the gene, or this side of the chromosome, or this side of the helix, which is in the 3 prime to 5 prime direction, and then I copy that to my RNA. And the RNA then becomes 5 prime to 3 prime, which is how we would then be able to transcribe then be able to translate. This is like another thing here. This is the transcription algorithm. If you have your input, your strand may be in the 5 or the 3 to 5 prime orientation. Your output then for the RNA would then be in the 5 prime to 3 prime orientation. So what I'm talking about here is that your DNA sometimes has to be reversed, sometimes it has to be complemented, and sometimes it has to be complemented and reversed in order for you to actually get to the gene that you need. Remember always that, I mean, don't worry too much about this right now, though, but remember always that the genes have a directionality. You have to start from a beginning on one side and work your way to the other side. And you have to ask yourself, well, which side is that? Am I starting at my proverbial left and working to my right? Or should I put this thing upside down and then start from the other side on the other sequence and work my way backwards? How do I read this gene? And that takes a lot of work here. And so these are just different ways of like looking at different kinds of algorithmic tools to determine which way the gene is able to be read. So remember that remember the directionality plays a very big thing. That's really the end of my slides here. One thing I wanted to do though is I wanted to spend some time just quickly to tell you that I'm updating the planning your time in this class. If you click on this, if you go to the class website here, you have in your planning your time, you have an idea about what we're covering, what's going on in this class, like what the basic ideas are. The light pinkish orangey colored stuff, these boxes here tend to have like the notes, like what are the overviews, what are the objectives of this class for that week. And then you have, for instance, the white boxes, those are classes, the blue boxes, or the blue entries here, those are labs. And so you can have an idea about looking at this, what we've been working on in our labs. And I'm hoping that this will help you to get ready for the exams when we have them, and I'll talk about this in the next weeks to come. But you'll be able to see, for instance, like where we've come as a class, and that will inform how you're supposed to be studying for this exam. Now I'm still going to put the other weeks in here, but I think we're basically caught up. And then another thing I wanted to talk about, by the way, are there any questions over any of this stuff that I've been mentioning so far? If there are any questions, I mean, please let me know. I don't want you to harbor any questions here when you can easily just ask what they may be. Okay, so the next thing I wanted to talk about though was you'll notice that in your directory there was a file called dogma. Let me just go ahead and show you something here if I can just get there. Oops, this isn't the right terminal. Hold on a second here. Let me just bring you to my terminal, and so you can see what I'm doing over here. That's not the terminal that I'm working at. I'm working at this terminal. So this is the terminal inside your. This should be where your class docs is. So this is your class docs lessons 08 to the 19th of March, cystic fibrosis source, and inside here we have dogma. This was the file that we began working on in class last time. I've added a few things to it. And I do hope that you spend some time here actually looking over this code to convince yourself that that this does work and to kind of get an idea about how it works. But really what we're doing here, let me just go over this quickly in the 2 minutes that we have left, is that we have, for instance, it just has it welcomes you into the program. This up here is just information that you need for that kind of talk about where the program came from, when it was made, and blah, blah, blah. This code up here, by the way, that's a Unix thing that allows you to run this code without having to type in Python all the time, but we don't have to worry about that. This is your main function here. That's your driver. First of all, it prints up some statements saying welcome to the program. Then over here, it assigns two different sequences, sequence A and sequence B, to these DNA sequences. Now, this is over here, this seq function is the library saying, I'll take that string that you're working with, and I'll convert this to my own so I can work with it more efficiently. And so that's what it's doing here. It's just converting the string to something so that it can work with it more efficiently. Next, what we're doing is we're just printing out the sequences, and then here we're figuring out the reverse, the complement of that sequence, and that's just done by taking the sequence name itself and putting a dot complement, because this is an object. We're able just to kind of like treat this string with this function. And the same thing is true here. We're taking this string here, and we're treating it with this function, or applying it to this function. And then in both cases, we're just printing out the output. And we do that for the complement, the reverse complement, and we also do that in the same way to transcribe this. And so in this DNA, or in this DNA analysis program that we just created here, we're actually able to create the RNA, the reverse complement, and the protein that comes from this DNA, which to us is not any more programming than just one line of code. But what we've done at the end here is that I'm actually now comparing these sequences. And by this, I'll just run this and show you what it does. Whoops. There we go. You can run this by typing Python 3. Actually, you know what? I need to be in my Docker container first of all. What I have done actually is I put my files. Oops, that's not it. It's not here. It's on my trash. It is going to be in my Docker bio. There we go. I need to run my container. Container is up and running. I can see my files. By the way, use of paths to get to my things here. And now if I just go ahead and type in Python 3, dogma, you'll see now that it compares the sequences by getting sequence A and B. It says, what's common between, it goes sequence or base by base. And if the bases if they're not the same, then it says, I found an error. I found an error. And so let me just show you what that code looks like for here, just so you have an idea. But really what it's doing is this is a for loop, and it assumes, by the way, that both the sequences have the same length, and that the same piece of information or the same base is present at each position. And if they're not, that means it's an error. So what it's doing is it's going through the first sequence A, and it's saying, okay, I'm going to look at what the character is, or what the base is at the position A at this index. And if I find that in that sequence A at position 1 or position 2, whatever it is, whatever I happens to be, if I find that position I is not the same in both sequences, if it's not the same, then I'm going to count my mismatches and say that's a mismatch. That means it's not correct, which was, first of all, I have to do this. I have to set this thing at 0. But then I say to myself that if they're not the same, then print out that there is a mismatch. And this is basically done by just managing strings. And so I have a string here, which is this not equals, which would then be shown between these two different sequence pieces here. Print out the characters of each sequence. And then we have this. This is a trick, by the way. If you take your variables and you put them in these curly braces in a print statement with an F in front of it like that, you can so that you can actually superimpose your variables. Oh my gosh, we're like 3 minutes over. I didn't mean to take everyone's time. Sorry about that, everyone. I can't see my clock from not how my screen is set up. But I'm going to leave it from there. When we come back on Monday, we're going to go back through and do some more work with. I think we'll do some other coding. I'm not quite sure exactly how that's. Well, yes, I think we have more coding to come. And so I look forward to seeing everyone Monday. Everyone have a good weekend. Friday, enjoy the weather. Outside, get some air. Enjoy the sun. Yeah, do it. Thank you, everybody. Take care. If you have any questions, let me know by Slack. I'm available.